Okay, so now we're going to talk about protein synthesis, and we're going to begin the discussion by talking about its initiation as well as how amino acids get activated because they have to be activated in order to build proteins. We're going to talk about the prokaryote and then bring in some differences for the eukaryote and also talk about some regulation at the level of eukaryote. So as we know, bacteria ribosomes are going to form a 70S, and our eukaryote counterpoint is going to form the 80S. Now, we always talk about ribosomes as being complete, but in actuality, in protein synthesis, they do not come together initially. The 50S and 30S are kept apart, just as our 60S and 40S of our eukaryotes are kept apart. So for protein synthesis, we basically have 4 stages. We have an activation stage, initiation, elongation, and termination. The activation goes back to our transfer RNAs and the activation of the amino acids in order to bind to our transfer RNAs. And then once that happens, you're going to have a series of initiation factors to bind those, to bring everything together to form the intact ribosome. And then you're going to have another set of elongation factors that will help to make the actual peptide, and then of course termination factors to stop everything. Now with respect to activation of amino acids, this is going to occur in the cytosol, and you're going to have a series of essential components listed here. We have of course our amino acids that are essential. We're going to have those amino acids be activated. That's going to be done by those amino acyl tRNA synthases. We're then going to have tRNAs that are going to be important. This is going to need ATP, and magnesium is always associated with ATP. So with respect to activation, you're going to have the carboxyl group of the amino acid be activated, and this is important in forming that actual peptide bond. And then you're going to have the actual bond form between the new amino acid and the information in the messenger RNA. Charged transfer RNAs, which is a transfer RNA attached to an amino acid. So that's what's referred to as a charged transfer RNA. So we have our amino acyl tRNA synthases, and these are what are going to connect the correct amino acid to the transfer RNAs. We're going to have 2 synthases that we're going to talk about. They're going to be class 1 and class 2. Now, the first step in the reaction for activating your amino acids is to have an amino acid, to have a transfer RNA, requires ATP and magnesium. And then you're going to get an amino acyl tRNA, release of your AMP, as well as an inorganic phosphate. And so this is showing the amino acid with the ATP, and this is going to lead to the amino acyl AMP. Now, this is what's going to lead to the synthases. Now, again, you have 2 classes of synthase. Class 1 is where you're going to have the amino acyl group transferred to the 2' carbon. So here's the 2' carbon shown here, check mark. And then on the class 2, you're going to have the amino acyl group transferred to the 3' carbon, shown here. And that's really the difference between the class 1 and class 2. Now, there is some amino acids that prefer class 1 over class 2, but we're not going to go into that much detail about these. It's just that the class 1 amino acyl transfer RNases, synthases, are going to transfer the amino acid to the 2' position, and the class 2 will transfer them to the 3' position. All right, so now we're going to have initiation. We'll talk initially about initiation in bacteria and then bring up initiation to eukaryotes. So with respect to initiation bacteria, you're going to have these essential components. Of course, you're going to have a message. You're going to have your ribosomal units, your initiation factors. This is going to require GTP, not ATP, and magnesium is going to be associated with GTP. You're also going to have in bacteria a formal methionine. Now, this N-formal methionine tRNA is unique to bacteria and is not used in eukaryotes. Every other methionine in the growing polypeptide chain will not be the formal. This is only for the start or the initiation. Now, the messenger RNA is going to initially bind to the 30S ribosomal unit of the initiating amino acyl tRNA, and then the larger ribosomal unit will bind. So let's look at this. So we have here our amino acyl site, which is this A site. We have our peptidyl site, and you can think of that as growing the peptide chain. And then we have an exit site, and this is all associated with 30S ribosome. So basically, A and P are going to be binding to those amino acyl transfer RNAs, and E is going to bind to the uncharged transfer RNA. So these have already completed their task, and you're going to release the transfer RNAs. So all organisms are going to have 2 transfer RNAs for methionine, and then this is going to be you're going to have one for that initiation methionine, and then the second transfer RNA is going to be for the methionines that are within the actual growing peptide chain. So with respect to our bacteria, we have our 30S subunit, and that's going to bind to initiation factor. So IF equals initiation factor. And so you're going to have IF1, IF3 that are going to bind. IF1 is going to bind to that A site, and this is going to prevent the binding of our transfer RNA. And then IF3 is going to bind to the E site. Both of these are also important in making sure that that larger subunit of the ribosomal RNA does not attach prematurely. Now, the initiation codon is going to use, again, that tRNA with the formal methionine for bacteria. That's the N-formal methionine. And again, this is only in bacteria. Now, you're also going to have here what's called the Shine-Delgarno sequence, and that's only in bacteria as well. And that's going to position the ribosomal RNA, the messenger RNA, so that the 5' methionine is available at that P site. So that methionine, that AUG, is going to be at that P site. Now, all other additions of incoming activated transfer RNAs are going to enter through that A site. But right now, that A site is still blocked with that initiation factor 1. So in the second part, you're going to have initiation factor 3 and messenger RNA joined by your GTP. So here you have your initiation factor 2 that is going to bring in GTP, and this is going to bring in your transfer RNA with the anticodon that is going to be, of course, pairing with the codon, and the anticodon has to pair correctly. And this is, of course, bound to that formal methionine, so that's that first initiation. Now next, you have the 50 subunit comes in, and this is going to initiate the release of these initiation factors. And it's going at the same, when this is happening, the GTP bound to initiation factor 2 gets hydrolyzed, and the GDP and inorganic phosphate will be released. All 3 initiation factors are released, and you are ready for elongation. Now, in eukaryotes, the methionine residues used are not going to be that formal methionine. And the initiation transfer RNA is going to be different than the one used for bacteria. Now, in eukaryotes, you're going to have a bit more complexity associated with this. You're going to have eukaryote initiation factors. That's what that E means. So this stands for eukaryote. and this is again initiation factor. And so initiation factor 1A and F3 are going to be very functional to that bacteria 1 and 3 we just discussed, which prevents the premature addition of the larger subunit to form the fully functional ribosome. So our eukaryote initiation factor 1 will bind to the E site, our 1A will bind to the A site, and then you have eukaryote initiation factor 3 binds, and this helps to prevent that 60S subunit from binding. Now what's going to happen is that you're going to have a charged initiator, and that is shown here. We have an initiation factor 2, which has that GTP bound to it. And this is going to form a complex, and here we have our first base coming in, that methionine base, and the complex is going to form what's called a 43S pre-initiation complex. And so in this case, you can see here that we don't have our messenger RNA yet. So we have no messenger RNA. And so this is different. We have no Shine-Delgado sequence. And so the ability of messenger RNA to find its way into this pre-initiation complex is going to be different. So before we start about that, we want to talk a little bit about the initiation factor 2 alpha kinase. So initiation factor 2 alpha, if you want to stop protein synthesis, one of the easy ways to do this is to prevent that initial methionine from coming in and binding. And so there are many conditions where you want to stop protein synthesis. Stress conditions, where you have a virus infection, hypoxia, you have UV damage. In those situations, you might want to be stopping because you don't have enough amino acids, you don't want your viruses to take over your cells, etc. And so all of these are going to have an effect on your eukaryote initiation factor 2 alpha. And so your 2 alpha, remember, your 2 is the one that's going to be coming in with your charged, sort of the charged initiation factor that's bound to the GTP. So if you block this, then you block protein synthesis. And so the phosphorylation of 2 alpha is going to help to bind to it our eukaryote initiation factor 2 beta. And then our beta is going to control our alpha, whether or not it's going to be able to switch GDP to GTP. So in conditions where protein synthesis is occurring off, you're going to keep the 2 alpha in a GDP situation. So you're going to turn off your global translation. You can also control the upregulation of genes that are more prominent in stressful environments. And so that's that ATF4 translation gets turned on. So you might want to undergo apoptosis. You might want the cell to become senescent or whatever it is. Now, but we still haven't added our messenger RNA. So this is where eukaryotes are different as well, because we don't have that Shine-Delgado sequence. We have a more elaborate sequence, of course. And so this is going to involve initiation factor 4F, or initiation factor 4 in general. So the 4F complex is going to include your eukaryote initiation factor 4E, and that's going to bind to the 5' cap. And then we're going to have initiation factor 4A, and that's going to have helicase activity as well as ATPase activity. And those guys are shown in here. And then we're going to have initiation factor 4G, and initiation factor 4G is referred to as a linker. So initiation factor 4G is what's going to provide a link between the messenger RNA and the actual 43S pre-initiation complex. 4G is also going to bind to the poly A tail, And 4G is going to wrap, sort of, you can see the cap is here and the tail is here. So it's going to wrap that tail around, circularize the messenger RNA. And the polyadenylation, it's the poly A tail interacts with the 5' cap. And you also are going to have the poly A binding proteins, and that's what helps to facilitate that binding to the 5' cap. So the poly A binding proteins are going to help with the circularization of your messenger RNA. And so now we have our 48S complex. So that's our 43 pre-complex plus our messenger RNA and all those initiation factor 4 proteins associated with it. We're going to have a scanning of our message for the start codon. So we're going to look for that start codon. And scanning is going to be facilitated by that helicase activity of our 4A. And our 4B. And then once you're going to find that starting codon, that AUG, your initiation factors get released. So all of these guys here and here, they get released after you find that start codon. And you're going to get hydrolysis of the GTP, and this reduces the affinity of that the initiation factor 2 for that tRNA that it brought in. So it kind of releases itself. At this point, you're going to be able to bind now that's 60S, and you have the complete 80S subunit that is formed. Now, as you can imagine, initiation factor 4 is also going to be a site where you can have regulation. Initiation factor 4E is actually expressed at the lowest level of all the initiation factors. And there are 3 mechanisms that are known to regulate this. One is at the level of transcription of the actual gene, so turning on the gene in the DNA. Another is modification through phosphorylation. And you can also have inhibition with those poly A tail binding proteins. So in summary, protein synthesis, of course, is going to occur on ribosomes, but the ribosomes don't come together until you have the messenger RNA in place. And after the messenger RNA in place and that first methionine is there, then in both prokaryote and eukaryote, you get the final ribosome formed. Every tRNA is going to have an amino acid arm with the terminal sequence CCA, and this is where your amino acid is actually going to bind to. The transfer RNA also will have that anticodon arm, and that's going to line up with the codon from the messenger RNA. 2 structural arms, the D and the T arm. And then you're going to have a growth of the polypeptide chain as you're going to begin with the terminal amino acid, adding to residues at the carboxy terminal end. Protein synthesis is going to occur in 5 stages. We talked about 4. The 5th stage is going to be folding into those actual 3D structures. But basically, you're going to need to activate the amino acids, you're going to need to initiate, and we're going to talk about elongation and termination.